Okay, so we continue to talk about the microbes, and after that brief introduction to different types of microbes, I wanted to discuss with you why we are in this class anyway, except for the fact that this class is a requirement for admission to different types of health profession programs. Okay, why we study them? Because these guys are everywhere. For me, probably the most unusual place where you can find microbial life is La Brea tar pits. I've never been to Los Angeles, but it's definitely, if I am ever there, that's one of the stops that I definitely want to make. So that's the site in Los Angeles where there are pretty much puddles of tar, molten tar. It was used by mafia in the 30s to get rid of the bodies. They were just dumping people, I don't know, alive or dead in those pits, and you can never recover the body from there. Turns out in that molten tar are microbes. You can find them in Mariana Trench, you can find them in the hot vents in the bottom of the ocean. I don't know. If you heard about Lake Vostok in Antarctic, it's the lake that's 2 miles under the ice. It was exposed on the surface something like 100 million years ago, and since then it was completely covered with ice. For like 100 million years, there are microbes there. Microbes that we don't know about. There's not a single species somewhere else on Earth. It's amazing. They are everywhere. Okay. Not only this, without microbes we wouldn't have normal geochemical or biogeochemical cycles. Decomposition, for example. Without microbes, Earth would be covered in the layers of dead bodies, human bodies, animal bodies, plant bodies, anything. Fungi and bacteria, main participants in decomposing the bodies. If you think about other cycles, nitrogen that comprises 78% of the atmosphere, only microbes can what we call fix nitrogen. Only microbes can convert nitrogen to the compounds like ammonia that plants can use to grow. We then can eat plants, because we still do not have a good chemical reaction to fix nitrogen. Microbes do it in the soil. It's a bacteria of the genus Rhizobium. Microbes fix nitrogen in the soil at the normal atmospheric pressure and room temperature pretty much. We have humans to make ammonia, built the plants at which ammonia is produced at the pressure of 300 atmospheres and the temperature of about 1,000 degrees Fahrenheit. So, we're still far behind. Okay. In many industrial applications that we do, microbes are absolutely indispensable. First and foremost, and I don't think anybody would argue with me, the first most important human application where microbes are indispensable is making of beer. Second, of course, goes wine. And third goes cheese. Okay. Well, then bread. And then we can talk about things like mass production of ethanol or making antibiotics. But beer is by far the most important part. Seriously speaking, if you use nail polish is acetone based. Acetone is produced by microbes. Isopropanol in a hand sanitizer is most likely produced by microbes, which is quite ironic because you use isopropanol produced by microbes to kill other microbes. Okay, so I think I kind of convinced that brewing and bread making and cheese making and wine making is all very important for humans. But nonetheless, the extremely important area of human life that's affected by microbes is health. So how many people die each year of the microbial infections? Now, let's take a look. The major killer in the world is heart disease. Second goes stroke, and technically you can put them all together because it's a cardiovascular disease, right? Stroke is the disease of vessels, so it happens, it's in the brain, and heart disease, also cardiovascular cause. Chronic obstructive pulmonary disease, mostly smoking, air pollution, not really infection. And then we go. Lower respiratory infections. What's the part of what is the lower respiratory tract? Actually, the whole lungs, trachea, bronchi. Okay? Lungs mostly. You know what the main infection of the lungs? It's the symptom. Tuberculosis kills the most. Pneumonia, actually, turns out pneumonia kills much less than that. Because we manage it really well. We have very good, effective antibiotics. Tuberculosis, especially in Africa, kills a ton of people. Infection of trachea, I don't know why they put it in a separate category. So technically, all lower respiratory tract infection kills about 5 million people. Diarrhea, 1.5 million. Now think about diarrhea. If you have diarrhea, is it going to kill you here in the States? Most likely not. Are you going to be able to work? Well, it depends on how bad it is, but most likely not for a day or two. Now imagine that we talk about millions and millions of people that experience that very unpleasant symptom every year. Economic losses are really hard to estimate, but they're huge. That make sense? Okay? So this is another aspect that I want to discuss with you that we often don't think about. We used to be scared of diseases like Ebola, right? Which killed 15,000 people over 1 year outbreak. It's not little, it's still a lot. The approximate number of people who are infected with helminths is about 900 million people. So those people live with a worm in the body. And these worms can really make their life miserable. They aren't going to kill them, but people will be partially incapacitated. Does that make sense? And that's economical burden. Anyone had common cold this year? Well, how was it? It was pretty, for me it was pretty miserable. Well, I had to go to work, but other than that, it was pretty miserable. And honestly, if I would work in some other place where, students wouldn't depend on me, I would just call in sick. I would just say, I'm not coming. Imagine millions of people in the United States every year call in sick because of the common cold. It's not going to kill them, no. They're just going to stay home. The economic losses are in tens of $billions a year. And the lost productivity, healthcare costs, the lost wages, all that stuff. There's a lot of indirect economic impact of diseases on the society. And you have to think about it, really. Because the scariest microbes they usually claim not really a lot of humans' lives. That's the minor quote-unquote infections that actually have a profound effect on our lifestyle, our economic well-being, and so on and so forth. Okay? Does that make sense? Now, before we move on with actual microbial features and lifestyles, properties. We need to learn how to classify the microbes. Since microbes are the part of life, they're going to be classified as a part of life, as a part of all living organisms. There are two systems that are used to classify life. One is called a Whittaker system, that is based on the observable traits, okay? Mostly on observable traits, mostly on the anatomical, morphological features, right? So for instance, if you would look, let's go into the animals, okay? If you would look at the tree of animal evolution, you will see that anatomically, what birds are pretty much feathered reptiles. If you would look anatomically at reptiles and mammals, you will see that both reptiles and mammals have evolved from amphibians. Okay, you can just look at the anatomy of the body. Does that make sense? So Whittaker based his system on that morphological evolution of species, and Whittaker, okay, isolated 5 kingdoms: animals, well, self-explanatory; plants, self-explanatory; fungi, self-explanatory; protists. Protists include unicellular eukaryotic organisms. It can be algae, it can be protozoa, those are all protists. And the 5th kingdom is Monera. Monera in Whittaker classification was based on Monera in Whittaker classification includes bacteria and archaea. Does that make sense? Okay, so we can put like morphological, evolutionary similarity. Okay. However, with the development of the tools to analyze the nucleic acid, the genomes. By the way, if I go too fast, tell me. Can you hold on, please? Can you say some? Can you repeat and stuff like that? I'm absolutely fine with that. Okay. So with the developing tools to establish the sequence, to see the sequence, to read the sequence of the genomes, the two scientists, Woese and Fox, they started to question, is the morphological similarity the best way to classify organisms? And Woese, what he was doing, this guy, he was isolating gene 4, so-called 16S ribosomal RNA, okay? So he isolated the 16S ribosomal RNA gene, and he determined the sequence of that gene and then compared two organisms, how the sequences are similar. Now, his presumption was, if sequences are very similar, the organisms are very closely related. Does that make sense to you? Like the farther the sequences are, the less similar the sequences are, the less organisms are related. Does that make sense? Think about this. Just think through. Okay? It's like with languages, right? Trying to think about something like, okay, what? French and English. Trying to think. Spanish and Italian. Good example. Okay? They closely related languages. Okay? And that shows that they probably arose from the same predecessor in its Latin. Okay? You look at like English and French, and you can see a lot of French words in English language or vice versa. Okay, they experienced a lot of influence on each other, so they're similar, they're related, historically related. The same goes for genome sequences. The closer they are, the more these organisms are related. Does that make sense? So Woese was looking at different bacterial species. Mostly he was trying to classify bacteria. And he acquired a sample of some microbe. And he sequenced it. And it just didn't make any sense. For him, absolutely not. Just no sense at all. It wasn't a bacteria, pretty much. It looked like a bacteria, it walked like a bacteria, and quacked like a bacteria, but it wasn't a bacteria. So he repeated the experiment and still got the same result. So it turned out he discovered a completely new domain of life. And so something that is absolutely different from any eukaryotes or any bacteria. He discovered archaea. Not in the sense that he was the first who found archaea. Does that make sense? You know what I'm trying to... He was the first who identified that this is something new. Right? You see what I'm trying to say here? So Woese proposed different classification based on the genetic similarity. Genetic relationship between the groups. He proposed the classification of life into 3 domains: bacteria, archaea, and eukarya. Why was so important? Because his method allowed to identify so-called horizontal gene transfer, the movement of genetic information from one group of organisms to another. Okay, from, say, movement of genes, and there is a transfer of genes from, I don't know, bacteria to humans. Okay, from humans to bacteria. There is a gene transfer, not just slow developing in a vertical manner during the evolution, but transfer of genes between species. And as I mentioned, he was comparing the sequences of the 16S ribosomal RNA. The sequences were conserved. What does conserve mean? Conservative. When something is conservative, what does it usually mean? Do conservative things like to change? No. So conservative in biology means it doesn't change that much. So if it changes, then it's a different species. Like we have, there are probably going to be a lot of genes in us that are not conservative that will determine why somebody has, different hair color or shape of the nose and whatnot. Okay, these genes are conservative. That's why Woese used them. So 3 domains of life, as we mentioned: bacteria, archaea, and eukarya. 2 of them are anucleated. 2 of them are prokaryotes. Okay, so these prokaryotic domains are bacteria and archaea. While eukaryotes, of course, do have nucleus. Okay. Now, viruses, as you can see, are not in the tree of life because they're not considered life. Any questions so far? Now, I want to erase this and show you something. So this thing that Woese and Fox developed is called phylogenetic tree, because it kind of looks like a tree. You follow? Now, based on that picture, can you tell me which 2 domains are closer to each other than to the third one? Look at the branches. Look at the trunk at the bottom, and then look at the branches. Two domains are genetically closer to each other than to the third one. Which two? Really? That's bacteria stems here, and archaea and eukarya come from the same branch. Do you see that? Bacteria separates earlier. So it turns out that genetically, archaea and eukarya are closer to each other. Okay, they're in more intimate relationships. If you think about it, it's something like you have brother and sister, and you have a cousin. Cousin is still related, but it's farther. That makes sense. Okay. So what do you have to know? You have to know which classification is based on what. Okay. You have to know 5 kingdoms. I'll ask, I promise. You have to know 3 domains. I'll ask, I promise. And you have to know the relationship between the domains that we just discussed. Okay? Now, moving on. How do we call the microbes? Except for the viruses, everything else has so-called binomial nomenclature. It's a universal way to name living things. Binomial nomenclature. First word is always for genus. Second word is always for species. It is a little confusing because generally we say, okay, lion, animal, lion, belongs to the genus Felis, and the species of lion is Felis leo. Leo is assigned to a species. Does that make sense? So in bacteria, you may have a genus of Bacillus, and the species is Bacillus anthracis. Second word determines the species. Does that make sense? It's like a make and the model of the car. Right? So, when somebody says they have, I don't know, a Honda, that's just a make. There are many Hondas. Okay? But people usually say, what do you drive? Honda Pilot. Okay? So they say make and a model. Same here. What is the species? Bacillus anthracis, Staphylococcus aureus, Clostridium difficile. Does that make sense? All staphylococci belong to the genus Staphylococcus. All bacilli belong to the genus Bacillus. There are different species of them. Does that make sense? Okay, I'm going to ask what these words mean. Like, first word for, and second word is for. So you have to know. Right? Now, classification and assignment of the names, it's really a complicated process, to put it the easiest way. There's a saying among microbiologists that there's a special place in hell for taxonomists, for people who name the microbes. They constantly change the names. Like it was, I don't know, something that used to be Pseudomonas can become Burkholderia overnight, because they decided it doesn't belong to Pseudomonas genus anymore. So they're moving stuff all the time. For viruses, there is a separate group called International Committee of Taxonomy of Viruses, and they classify viruses based on the replication cycle and the genome structure. So far, you have to know the binomial nomenclature. Am I clear? Now we're going to go back to like first grade. Now this is going to be an exam, but it's more of a review. I want us to be on the same page in terms of the chemistry of life. Okay, so first of all, no water, no life. Humans are what percentage of water? Well, that's probably if you like drink a whole jug, 6 to 7, around this. Bacteria, almost 100. So bacterial cell is almost all water. Okay, there's a lot of water in there. Now, in the water, few things that we have to know about it. First, water forms hydrogen bonds. That is why water is liquid. By all other chemical laws, it should be gas, but instead it is liquid. It is liquid because oxygen pulls electrons, okay, from hydrogen. When oxygen pulls electrons, oxygen becomes partially negative and hydrogen becomes partially positive. Positive and negative attract, there you go. So you have like partially positive hydrogen, partially negative oxygen. They will attract to each other. So water molecules will be attracted to each other. Does that make sense? This attraction is called a hydrogen bond. Hydrogen bonds are extremely important in shaping the biological molecules like proteins or nucleic acids. For instance, the double-stranded DNA, it is double-stranded because two strands are held together by the hydrogen bonds. Okay? How can we destroy the hydrogen bond? Hydrogen? Go ahead. Okay. Let's think it through. Hydrogen bonds hold water molecules together so the water stays liquid. If hydrogen bonds disappear, what's going to happen to the water? So, how you destroy hydrogen bonds? You heat it up. Does that make sense? And that is how you destroy hydrogen bonds in water, in biological molecules, everywhere. You heat it up. That makes sense? Actually, formation of hydrogen bonds allows the ions like chloride ion and sodium ion to be dissolved in water. Okay. Partial negative charge of oxygen, is attracted to the positive charge of the sodium. Does that make sense? So keep in mind hydrogen bonds in water. Second thing is pH. Let's be very clear. All you have to know about pH is that when environment becomes more acidic, what happens to the pH level? It goes down, not up, down. Okay? I will not buy the excuse, you know what I mean. I know what you mean, but it goes down. So don't convince me, okay? Don't try to convince me that increased acidity means increased pH. No, it goes in opposite direction. So increase alkalinity, higher pH. For our purposes, acid means what ion? Hydrogen ion. And base means hydroxide or H. Okay? Just to be on the same page. Good? Clear? Okay. No, I will not ask you what is the pH of tomato juice. I don't really care. Okay? major molecules of life. We're going to start with carbohydrates. Carbohydrates consist of carbon, hydrogen, and oxygen atoms, and follow the empirical formula like this: CH2O taken n times. For example, the carbohydrate that you are all familiar with is the glucose. Okay, so this is the glucose here. Glucose, galactose, fructose are monosaccharides. So the example of monosaccharide, I mean, if you will try to kind of, just remove this. So one molecule of glucose is the monosaccharide. Does that make sense so far? Monosaccharides can form chains. Okay? They can form chains in the reaction of dehydration. So you have one monosaccharide and you have another monosaccharide, right? The water is taken away from them, and what you end up with is disaccharide. Does that make sense? Water goes away. Does that make sense? Everybody gets it. So this is why it is called dehydration. You take two monosaccharides, you take water out, they form disaccharide. Does that make sense? I will not continue until you tell me it does, or it doesn't. You, by the way, you're more than welcome to tell me, look, I have no idea what you just said. Can you explain it some other way? Right? You good? Now, obviously, if you will take more than two monosaccharides, you can build a long chain. Like amylose, or amylopectin, two components of starch, or glycogen, it's component, the polysaccharide, they're called polysaccharides, okay? Polysaccharide that can be found in the muscle of the mammals, right? Now, if you need to break it down, if you need to break down starch into the individual single into monosaccharides, which reaction you're going to use? Look here. Look at the reaction that we just wrote down. You need to add what? Water. Yeah, you took water away, you need to add water back. This is called hydrolysis. Okay? Hydrolysis is always catalyzed by the enzymes. Think about this. If you mix starch and water, what's going to happen? I mean, in thousands of years, yes. Like, in your lifetime, especially if those sterile, nothing. But, if you mix starch, water, and throw in some yeast, you're going to get alcohol. Because yeast is going to break down starch, okay? Release monosaccharides, and use them to produce alcohol. One of the great examples how water and enzymes act on starch. If you take a piece of bread and you chew on it for a very long time, you're going to feel a sweet taste in your mouth. The reason for it, starch in the bread is hydrolyzed into the glucose by the enzymes in your mouth. Does that make sense? You just have to take it. It takes a while. So what do we need them for? Sugars, carbohydrates. What do we need them for? Energy. First and foremost, energy. Second, structure. They are components of glycoproteins. Okay, there are components, important components, different monosaccharides are components of the bacterial cell walls or external covering of the cell. You can say that they are needed for the storage of genetic information. Okay, RNA and DNA have ribose sugars. Just checking that it's recording. They have ribose sugars as a part of a molecule. So you see, but the main and most and foremost is, of course, the energy. Any questions? So you have to understand the difference between mono and di and polysaccharide. Okay, the reaction, dehydration to make long chains, hydrolysis to break down long chains into the single molecules back again, and the main function. Good? Questions? Lipids. There are several types of lipids that can be found in the microbial cell, in a living cell. One is triglycerides. Triglycerides are composed of the glycerol and fatty acids. So you can see fatty acids here, and you can see glycerol here. Again, you combine glycerol and fatty acid, you remove molecules of water, and you get triglyceride. So again, this is the reaction of dehydration, right? How you break down triglycerides back into the elementary building blocks, into the glycerol and free fatty acids. Through what reaction? Hydrolysis, there you go. So it's going to work for practically all molecules of life. The main function of triglycerides is energy supply. Okay, microbes can use it. Practically all cells can use it. Right? Did that make sense? Okay, phospholipids. These guys are composed of the phosphate residue, glycerol residue, and fatty acid residue. Now, if you look at this molecule here, you will see that the head of the phospholipid molecule, the glycerol and the phosphate, they're hydrophilic. Okay, they can be easily dissolved in water, while the tail, the fatty acid tail, is very hydrophobic. So this molecule is called amphiphilic. Okay, it means that they have both, say, one part likes water, another part likes lipids. Where are you going to find phospholipid molecule in the cell? The membrane, very good. So the surface of the membrane, whether it is the surface that's exposed to the outside of the cell or the surface that's exposed to the inside, so either surface of the cell, either surface of the membrane is going to be hydrophilic. Why? Not yet. What? Think about microbe. What's inside of the microbe? What fills the microbial cell? Cytoplasm. What is cytoplasm composed of? Mostly water. So inside is the water. And if microbe is living, outside of it is something watery too. They live in the watery environments. So you have to have something that will not repel water on the surface. But if you have only something that is water loving, it will simply dissolve. So you need another component that would hold it together. And that is the hydrophobic portion. Does that make sense? Now, going back to transport. How will it determine the transport of stuff across the membrane? Say again. What will they do? Oh wait, well, for what type of molecules? You know what I'm trying to say? Like, what can something cross the membrane easy without any proteins, just directly through the membrane? Huh? Yes, what? What can? Yes.
Small molecules.
Like water? They gotta have another property. Not only they should be small. What is inside? What is in the center of the membrane? Like this layer. Is it this layer? Is it hydrophobic or hydrophilic? Hydrophobic. So can water-soluble molecules cross it? No. Which molecules can? Fat-soluble. So small fat-soluble molecules can go directly. Actually, not so small. There's a certain limit size-wise, but like ethanol can cross membranes pretty easily. Okay? Does that make sense? Now, for water-soluble, you gotta have some sort of the protein, the transmembrane protein, which we're gonna talk about. Right? Now, the third class of the fats, the sterols and hopanoids. You can see that they're fairly similar in structure. They have, they remind of each other. So sterols, like this cholesterol molecule on the left, they are parts of the cell membranes in the eukaryotes, exclusively eukaryotes. And they regulate how fluid the membrane is, how rigid it is. Does that make sense? Okay. Hopanoids, which can be found, in bacteria, they perform the same function for bacteria. So functionally they are the same, they have slightly different structure. Okay? So know the function, know what they are made of. And by now you should appreciate, hydrolysis and dehydration, the roles of those reactions in the synthesis of the biological molecules. Okay? Proteins consist of amino acids. Amino acids, some of the examples here on the right, they are linked together by the so-called peptide bonds. Okay? Again, peptide bond is formed as the result of the dehydration reaction. Right? Now, amino acids are different. They have different side chains. You can see that, for instance, side chain of lysine is absolutely different from the side chain of glutamine, aspartate, or cysteine. They all have different side chains. Okay? And these side chains, they actually determine the properties of the amino acid. For instance, alanine would be hydrophobic amino acid, while serine is going to be hydrophilic. Now, these side chains of amino acids will eventually determine the structure and the function of the protein. That's some other digression that I want to make really quickly. Structure and function. Are they linked? Yes. Yes. Period. Yes, they are. Okay. So, we can't even say that structure determines function and vice versa, but they intimately linked. Okay. if you would look at the wings of the butterfly and of the bird, they look fairly similar, okay? So they both are needed to fly. So function is why the structure is like that, okay? You're not going to see a wing that is shaped like, I don't know, like a leg of an elephant, okay? Just because it's not going to work, right? Look at all different types of wheels that we have. They all have the same function, so they have the same structure, right? So link between function and structure is very intimate. And of course, if the protein assumes the certain structure, which is determined by these side chains, it assumes a certain function. Proteins have several levels of structure. First level is a sequence of amino acids. Okay? It's your first structural levels. It's called a primary protein structure. Okay? Now, since amino acids have those different side chains, this initial structure will fold in a certain way. That requires a certain leap of imagination. Can you imagine that? Think about it. Based on which parts you put together, okay, it's going to have different shape. Does that make sense? So this shape is called secondary protein structure. Okay, there are two major elements: alpha helix and beta sheet. And then those beta sheets and alpha helices, they will fold further, okay, forming tertiary structure. Many proteins, tertiary structure is the last step. You know, if that's one protein molecule, that's it. However, some proteins, they actually start to, assemble together. Some proteins, they have multiple subunits. Okay? One protein that you definitely know about is hemoglobin. It consists of 4 subunits. So it has quaternary structure. Does that make sense? And what will happen? So, and these structures, the secondary structure, the tertiary structure, they are held together by hydrogen bonds. Not only, but predominantly hydrogen bonds. So, how can you break them down? Heat them up? Yes. Do you heat up proteins in the morning? Huh? When you do what? Exactly. Well, you cooking practically everything. You heat up proteins. You denature proteins. Every time you cook something, you denature proteins. And you can see the changes in structure. Egg in the beginning is what? It's liquid. You put it in the skillet, it becomes solid. Right? You denature proteins. Does that make sense? So, when protein that was doing something, it had certain structure, and you denature it. So change the structure. Can it perform its function? If you change the structure, not anymore. The function's gone, right? Does that make sense? Why I spend so much time on this? Out of those 3 hand hygiene compounds that we used, chlorhexidine and ethanol, can both denature proteins. And when bacterial proteins are denatured, bacteria cannot do stuff. They just cannot leave. They die. Okay, like if you heat up the human will die because proteins will denature. You know why? What is the highest body temperature that a human can survive? As far as
can survive not very high fever, like 106, but you're going to have brain damage. But like anything over that, technically your brain dead.
Well, one of the reasons your proteins start to denature. Because if it's temperature high enough, proteins just start to denature. That makes sense? Yeah, absolutely. Yeah, you're going to die. Brain damage. Now, nucleic acids. We have two types, DNA and RNA. And DNA and RNA actually both consist of what? A, T, G, and C. What is the common name for them? Nucleic acid consists of nucleotides, yes, of nucleotides. Okay, and remember, those nucleotides are complementary. A, T, G, C. This is the fundamental property of life that allows us to make copies of DNA molecule without losing any information. And since we have double-stranded DNA, bacteria have double-stranded DNA, all cellular organisms have double-stranded DNA. It means that genetic information is stored in both strands. Does that make sense? So you have a sequence. I always compare DNA to a book. It's the cookbook, your family book of recipes. It is stored in a precious place in some sort of a bookcase, maybe even locked. Okay, it's like 200 years old. It contains recipes that you have made up, your mother, your grandmother, your grand-grandmother, and it goes generations and generations before. Some recipes probably don't mean a thing anymore. You just cannot make them. Some of them are just pure gibberish. Because maybe when you were a 3-year-old, you just scratched parts of them. Just for fun. Okay? But it's cherished anyway because that's your heritage. So DNA is like that cookbook. So you may have like 30 different recipes for apple pie. And 50 for, I don't know, roast beef. Okay? Does that make sense? Think about this, because we're gonna come back to that analogy later. Now, the strands of the DNA can be copied for the cell division, because when cell divides, you have to pass on the genetic information. So you have to copy it every time. So genetic information will be passed on. Say, going back to that analogy, if your brother moves to another state, brother will make a copy of the entire book and take it with him. Right? Now imagine that you need to make a dinner. You have this thick of a cookbook. So what you're going to do? You need to make a dinner. You have a cookbook. What you going to do? Like the entire thing? Yeah, just what you want to make. Okay, so you decide maybe you'll make a salad, an entree, and dessert. Okay, so pick them, but then think about this. It's a big book, right? Are you going to carry it into the kitchen? That's your family heritage. You're going to carry it into the kitchen? Like the entire thing, 5,000 freaking pages. What you going to do? Yeah, well, I would actually use my cell phone. Probably just take a photo of the recipe and bring it to the kitchen. Okay? Same happens in the cell. Cell doesn't hold DNA back and forth all over the cell. Okay? In the cytoplasm, ribosome, everywhere. No. DNA is precious. It stays in the nucleus. Or if it's bacteria, it stays in the nucleoid. If cell needs to make something, and this something is going to be a protein, right? Cell expresses the information from the DNA in the form of messenger RNA. Again, it's all based on complementarity. So if you have DNA sequence of this, then RNA is going to be what? A is complementary with what? If it's RNA, it's going to be U. T is complementary with A, G, C, T, A. Okay? So it's absolutely guaranteed way to read the genetic information. Does that make sense? Think about this. Think about this step, the gene expression. DNA into mRNA. And mRNA is then going to be translated into the protein. Because everything in your cells, everything in the bacterial cells is done by proteins. Proteins are enzymes. Proteins are parts of the transport systems. So proteins build things, proteins make things, proteins facilitate things, proteins signal. Does that make sense? So you must end up with a protein. There are a few exceptions, but only a few. Now, said that word, gene expression. Let's talk about it a little more. So we figured DNA can be complementary, sort of copied, used for making RNA. And then RNA can be used as the template to make proteins. Does this process make sense to you? Just take your cookbook, snap few shots of, different recipes, then go to the kitchen and make a few meals. Those meals are the proteins. The snapshots is messenger RNA. The cookbook is the DNA. That makes sense. The first step in DNA is used for RNA synthesis. It's called transcription. And when RNA is used for the synthesis of the protein, it's called translation. Okay? So genome, essentially, your cookbook, genome, carries information about all the traits of the organism. All of them. Genome is the assembly, assembly of all the genes that you have. All of them. Genes for eye color, skin color, height, body buildup, your immune system and whatnot, okay? And when genes are expressed, genes of the genome, then they define the what? Genotype defines phenotype. Can you explain me what is phenotype? I want us to be on the same page in that too. Do we have different phenotype? Why? No, not the why. I mean, how it is different.
Combination of genetics.
Simpler. I'm male, you're female. We have different height, we have different weight, we have different body composition. We're different. Phenotype is appearance. Does that make sense? Okay, that's phenotype. Now, what would be the phenotype for bacteria? If one bacteria grows faster than another, is that a phenotypical trait? Yep, absolutely. If I grow faster than somebody else, that's my phenotype. Okay? If one bacteria is more pathogenic than another. Sure, absolutely. If one bacteria can metabolize lactose and another cannot, that is phenotype. Any functional difference, any difference in appearance, it's a phenotype. Does that make sense? And all those differences are defined by the differences in genotype. And you have to appreciate that expression of genes can be changed in response to the various stimulus on the outside. Okay? It's often easier to talk about humans. Okay? When I'm sick, when I have a common cold, what do I have? A runny nose. So I produce a lot of mucus. Okay? I produce a lot of mucus and proteins that increase inflammation. Expression of the genes changed in response to the stimulus, the infection. Okay? And my phenotype actually changed. Does that make sense? Okay. One very important thing, not all genes are expressed all the time. That's absolutely fundamental, and it's true for all organisms alike. Think about this. How many genes does human have? 20,000. We have 20,000 genes. We express percents of this number. Some genes are needed when we're cold. Some genes are needed when we're stressed. Some genes are needed when we're sick. Okay? So the regulation of expression is critically important. Same goes for bacteria. You know, bacteria are not going to express genes, all the genes that they have all the time. Right? You take E. coli, the most common. You've all heard about E. coli, right? The bacteria. It has, God bless you, 5,000 genes, something like that. It would be a waste of energy to express all of them at the same time. Okay, so that's very important. Now, one of the great examples of the genes that are expressed not all the time, okay, is the genes for antibiotic resistance. So there can be multiple mechanisms. One of the mechanisms, for instance, to expose microbes to the antibiotics. And if so happens that one microbe expresses the genes at a given time, genes that confer that resistance, it will outgrow microbes that do not express them at this time. Okay? And this change in the environment will make that expression required for the survival. So the gene will become what we call constitutively expressed, constantly expressed. You remove the antibiotic from the environment, there's a good chance that the expression of the gene is going to go down. Does that make sense? Okay? So that's kind of reflected here, okay, on this flow chart. In response to different environmental conditions, microbes will show different phenotypes, and these phenotypes will be defined by the gene expression. Right? Now, microbes evolve, unfortunately. They adapt. They not only change the expression of genes that they already have, they change them, they acquire new genes. And they do it through the mutations. Okay? Mutations are the changes in the genome of the microbes. Okay? And that confers the critical mechanism to adapt to environmental changes. Right? Great example is resistance to antibiotics. If a microbe has a gene that confers antibiotic resistance, you can see the same picture here. Okay? Microbe that has the gene that is expressed, that's important, gene that is expressed will survive in the environment that contains antibiotics, and eventually you will have the population of microbes that are resistant to this antibiotic. And that's what we observe now. Have you heard about MRSA? It made quite a splash about 15 years ago when it first rose, methicillin-resistant Staphylococcus aureus. It contains genes that give it a protection against methicillin. Well, actually, most of the antibiotics of penicillin group. So for a long time we thought we practically screwed, we need to treat it. The general sentiment now among clinical microbiologists, it's a new normal. We just have to accept the fact that Staph aureus is resistant to penicillin. Penicillin, methicillin, amoxicillin, ampicillin, so on and so forth. So we need to find new ways to treat it. So it happened, we selected it, okay? Does that make sense? Now in terms of evolution, there are a couple of thoughts for you about evolution. First of all, evolution is not directional. Do you understand what I'm trying to say? We do not ask why humans have 2 arms, 2 legs. We do not ask this. You know why? Because those who did not have the same phenotype, who were different, what happened to them? Why? They didn't survive. We did. We're lucky. Okay? It's not that we evolved something. We developed something. It's just under certain selective pressure, that's the word that I want you to know, selective pressure. Under certain selective pressure, certain phenotypes survived. Does that make sense? Think about this. How many species, how many genera, orders, classes disappear from the face of Earth because environment changed? I think it was Permian extinction. I think it was Permian. 90% of species on the face of Earth disappeared. Think about this. 90%. It's the largest extinction by far. Dinosaur extinction was merely a blip. Seriously, it was just nothing. Okay. So, same goes for microbes. They adapt. They adapt much faster. Why? Why they adapt much faster to the...
They're simpler organisms, so there's less complex processes that
Okay.
relate to new environment. So the simpler something is, the quicker it adapts.
Another reason. That's right. That's right. The simpler, the easier. Another reason. Oh yeah, they reproduce fast, so they can accumulate the changes faster. Does that make sense? So, now this statement, I really like it. Nothing in biology makes sense except in the light of evolution. When we talk about anything, we're going to look at it through the lens of evolution and gene expression. Okay? Only in this case it's going to make sense. So I'm going to bring up these 2 ideas a lot. Right? That makes sense? The question, what you have to know, link between what is genotype, phenotype? Okay, how are they linked? The idea that not all genes are expressed all the time. And that gene expression is what defines the phenotype. Right? So, I'm going at a very good pace. Okay. On the before we move on, you're going to receive your first quiz today. You bring it next day, and you bring both the scantron, which is what I grade. So these guys are graded, okay, and the quiz. Quiz is yours. I don't need them. But we review the quiz. We go through the questions and we figure out why the answers are these. Does that make sense? I usually tell students that it's not an open book when I give them home. which causes some sort of a confusion. Of course, it's an open book. Try not to Google. My personal suggestion, try not to Google, okay? Really. Not because, you know, you're going to find exactly same questions, but because when you... Huh?
You'll find so much information.
Well, let me be very blunt here. Google is lazy. Google gives you the instant answer, okay? If you use the PowerPoint, if you use your notes, you have to search for that answer. And you actually, turns out, you learn a lot while you are searching. Just kind of read it through. Does that make sense? So use your notes, use recordings, anything, but just work with the material instead of using Google. Does that make sense? Okay, let's chat about microscopy. That's going to be our major tool in the class for quite a long time. One of the major tools. So, what do we use microscopy for? To see microbes. To make them look what? Bigger. Right? So this is, we can do that by utilizing two approaches. Refraction and reflection. So essentially, if you will ever try to disassemble the microscope, what you're going to find inside is the bunch of lens, mostly convex, okay, and no, sorry, you will find mostly concave lens. Yes, convex is for the different type of optics, mostly concave lens, and little mirrors to direct the light in the proper way. It's just a bunch of lens. And the idea is that in the end, those lenses make small objects like this piece of cork bigger. So Robert Hooke was the first guy who designed the microscope fairly similar to what we have now. And he first actually saw cells. He never paid too much attention to it. He never realized the significance of what he saw, but he was the first one to see cells. The first guy who actually discovered, no, not discovered, you can discover the microscope, who made microscope, was Antoine van Leeuwenhoek, the Dutch glassmaster. He was making glasses for people. And one day he decided just to put several lenses in one tube and look at it. Well, guess what was the first sample that Leeuwenhoek looked at? He was not a scientist. He was a tradesman. He was curious though, very curious. His own crap. No, seriously. He spread it and he looked at it. And he was the first one to see protozoa in helminths, because it was 17th century and oh boy, people had a lot of that stuff in their feces. Okay. So, microscopes evolved, of course, a lot. You can see those. We're going to use those in the cabinet. They called light, because we use, not because they really light, they're actually heavy. Light because they use light to project through the specimen. Compound, because there are multiple lenses. Binocular, because there are two eyepieces to look into. Parfocal. Parfocal means that when you change objective lens on the microscope, the picture remains in the relative shape. Relative focus. Relative focus. Does that make sense? I mean, you have to adjust it a little bit, but it's still okay. That makes sense. So there are a few terms that you must know about the microscope. First, obviously, is magnification. Magnification is simply how bigger the object looks in the microscope, and it is determined by the magnification of the eyepieces and magnification of the objective lens. You multiply one by another. So for instance, eyepieces on those microscopes and on mine, magnification of eyepieces is 10. So if I put a specimen under 20x objective lens, what's going to be the magnification? 10x here, 20x here. They should be, look at this, one by another, one multiplied by another. What's the magnification? 200, 20 by 10, 200, right? That makes sense? So the microscopes that you're going to work with, they have 4 eyepieces, 4 objective lens, I'm sorry. They have 4x objective lens, 10x, 40x, and 100x. So you can observe your specimen through with the magnification of 40, 100, 400, and 1,000. So they're pretty good microscopes. Right? Good. Second feature is the ability to see the fine details of the specimen. We call it resolution. It is determined by the wavelength or same frequency. Okay? Wavelength and frequency are kind of an opposite parameter. The higher the frequency, the shorter the wavelength. Does that make sense? Now, the higher the frequency, the better the resolution. Okay? Light with the shortest wavelength will give you the best resolution. So essentially, it's going to be blue light that gives you the best resolution. Now, this is also what puts the restriction on the size of the object that we can observe under the light microscope. Okay. It's practically, it comes from the physics. I know physics is your absolutely favorite subject as well as mine. I know that. It's quantum physics that determines the smallest specimen, the size of the object that can be observed with a certain type of microscopy. For light microscopy, the smallest is about 0.2 micrometers, which is really small. Okay. What if you want to look at something smaller? What do you have to reduce? Yeah, so you essentially have to go down in terms of the wavelength. You can use electron microscope in some cases. For some applications, it's not technically microscopy anymore. It's not light, but you can sometimes use UV light if you have a way to detect it. You can shine UV light through. Okay. Some applications use even gamma rays or X-rays for certain things to determine. That makes sense. Okay. And the third parameter that is very important for the microscope is contrast. Okay. So essentially, you always want your picture to be contrast. The brightness of your sample and the brightness of the background should be different. Okay. You don't want to observe gray and white. Does that make sense? You would rather look at red on white or blue on white. You see what I'm trying to say? Contrast is very important for the good sample observation. We're going to talk how we increase the contrast. So again, before we move on, what you have to know. I don't really care about Hooke and Leeuwenhoek, they dead. But contrast, magnification, and resolution, right? Know what they are. If I give you like this, blah blah, this feature of microscope is how bright the sample is regarding relatively to the background. That's going to be contrast. Okay. Ability to distinguish the fine details is resolution. Know how we determine the magnification. Okay. Parts of the microscope. You see that microscope? My microscope is much cooler than yours. You know why? Because I'm much cooler than you. Just kidding. No. Actually, the big difference between this microscope and yours, there are two of them. One, it is hooked up to the computer, and sometimes it works. It's also kind of magic. I mean, half of the last semester, we just couldn't put the picture from the microscope on the computer. Technology. And the second, it has a little bit more options in terms of the specimen observation, right? Yours is a straightforward light microscopy. I have a couple of phase contrast, which produces a little bit better image. It's a little bit more sophisticated. Other than that, you probably will work with that type of microscope in the clinic if you ever will. Okay. So, what are the main parts? Well, eyepiece. This is where you look into. You don't look like this. You won't see anything. You have to find the perfect spot a little away from the ocular lens. Okay? You will see one window. Okay? Does that make sense? Objective lenses. Here. They are placed on the revolving nose piece. This is called nose piece. Can everybody more or less see it? Okay? You always start with the what magnification? Highest, lowest, in the middle? Lowest. Always start with the lowest to locate your specimen. Okay? So, in my case, it's actually very inconvenient. In my case, the lowest magnification for the microscope is going to be 100. For you, it's going to be 40. 40 is way better because you can see a bigger chunk of the sample and you can locate things quicker. Okay? So you start at this lower magnification, which is called scanning magnification or scanning objective. Okay? And you bring it in focus. To bring it in focus, you use the coarse focus knobs. How am I supposed to do that? Okay, hopefully it will not fall. Okay, can you see the coarse focus knobs here? The big guys, okay? They move the mechanical stage. You can see the mechanical stage here. Okay? They move the mechanical stage, the coarse focus knobs are here. They move the mechanical stage a lot. So they help you to focus on the specimen, the very, very low magnification. Right? And once you got your specimen in the proper focal plane, okay, you can move on with magnification. How you determine that your specimen is in the focus? How do you know that it's actually a specimen and some artifact? Unfortunately, I have only one answer. Common sense and experience. First of all, you can have a lot of bubbles. Don't let them trick you. That's number one. Okay? So, they pretty discernible. Second, when you focus your specimen, move your stage forward and backward, left and right, a little bit. You see those mechanical stage knobs? They hang in on this side of the microscope. They allow you to move the stage. Now, if whatever you look at is moving, it's the part of the sample. Does that make sense? If you look at something, you move the stage, and it doesn't go anywhere, that's the dirt on the objective lens. Disregard it. It's a wrong focal plane. Does that make sense? Okay. Once you go from the coarse focus knobs, once you go from the lowest magnification to the next one, you stop using coarse focus. Use the fine focus knobs. So you elevate or bring down your stage by very small steps. The reason for that, bring it up, you're going to crush either the specimen or the objective or both. Specimen is fine, but objective, they are expensive. We don't want that. Right? That makes sense? Now, in order to look at the sample, you have to actually turn on the light. You're going to find their buttons. Look, it's on-off button. You can regulate how bright the light is by little dial here that adjusts the light intensity. You have an element that is called condenser that is somewhere here. You don't really care about it because what condenser does, it focuses the light on the specimen, small spot in the specimen, right? And you have diaphragm. That's the important thing. Those of you who do photography, you know that by changing the diaphragm, you can change the contrast of the picture. Same goes for microscopy. When you have your sample focused, by carefully playing with the diaphragm, it's a little, I don't know how to explain it. You're going to see it, I don't have that microscope, it's a little dial under the mechanical stage. You can adjust the contrast. Does that make sense? By making it a little darker or a little lighter, you can change the contrast. It'll help you to observe the specimen. Again, I'm giving you an overview of the parts of the microscope, but until we actually get to the microscopy, it's not going to make a lot of sense. You really have to touch it, and you will touch it. Okay? Now, we're going to wrap it up now, because I don't want to start like a new topic. When do we finish? 11:50 or 11:55? 11:55. Then we'll talk about light microscopy. I thought 11:50 for some reason. Okay, we're going to talk a bit about light microscopy, a few things. So we're going to, what I want you to know about microscope, before we move on to this one, parts, what they do. Am I clear enough on that? Okay. Now, types of microscopy. Light microscopy uses light. And the type that we're going to work exclusively, almost exclusively, so-called bright field microscopy. In this case, you observe the darker specimen on the bright background. Does that make sense? You see why it's called a bright field. You have bright lit background, and the specimen can be colored, may not be colored, but the background is bright. It's the most common type of microscopy. In the dark field microscopy, okay, in this case, the background is dark, and the light that is refracted in a certain way lights up the bacterial cells. So these bacterial cells start to shine on the dark background. It's very convenient for detection of really thin bacterial cells, like microbes of the family Borrelia. Genus Borrelia. Borrelia burgdorferi is the causative agent of Lyme disease. Or microbes from the genus Treponema, which one of the representatives of Treponema pallidum, is the causative agent of syphilis. Dark field microscopy is the common method to detect either Borrelia or Treponema. Does that make sense? Phase contrast. The great British sci-fi writer Arthur C. Clarke once said that at a certain level of development, technology is indistinguishable from magic. So for me, everything that goes above dark and bright field microscopy is magic. It's a level of technology which I can use, but I have no freaking clue how it works. It's like a computer. I have no idea why and how I can do pictures here, okay, using this pen, and it all gets recorded and goes on YouTube. I cannot wrap my head around it, but that's fine, as long as it works. Right? So, phase contrast. There's some manipulation with light, with interference and diffraction and refraction. What you end up with is the live specimen, which is of a, image of a live specimen, often live specimen, high contrast, high resolution. This microscope can do phase contrast, and when we will observe microbes, if I can put it on the screen, you will see it here. If I cannot, you're more than welcome to come and take a glance. It looks pretty nice, actually. The individual cells, very high contrast, swimming around. Okay. DIC. Again, increasing contrast and produce more of a three-dimensional picture. Okay. It allows you to appreciate the shape of the microbe to a certain extent. Does that make sense? Again, it is all based on the interference of light. Okay, it combines different wavelengths, or it emits light at certain time points, so the waves can interfere. Okay, very detailed structures of the specimen. Fluorescence is pretty simple. So, fluorescent microscope uses what we call a black light, right? So, you have a specimen that either can fluoresce by itself, or you add some kind of fluorescent dye, fluorescent chemical, that will help cells to fluoresce under the UV light. Does that make sense? It's like, have you seen like the t-shirts that start to glow in the dark stuff? That's black light. That's pretty much the same principle. You have a special dye, you put the light on and the dye starts to fluoresce. If you make the dye specific to a cell in some way, you can detect only one type of cells, but not another. Does that make sense? Confocal. Let me finish with this. Confocal. You're familiar with tomography. When you do tomography, you take slices, right? Just take slices, like you produce image in different slices of the human body, so you can recap the 3D structure. Same goes for confocal microscopy. Microscope takes slices through the specimen it changes the focal distance and takes slices through the specimen, producing essentially a 3D picture in many cases, not in all cases, but in many cases. It gives you an appreciation of the shape because it takes different slices. It's all computerized now. It's not that you manually change anything. Okay. 2-photon. That's fascinating. I have no clue how 2-photon microscopy works. What I can tell you, it is very convenient to work with specimens that are thick. So you can actually have a slice of tissue and observe bacteria inside of the slice of tissue. You can use live tissue like a thin muscle. If you can put thin muscle under the microscope and if it's live and there's blood flow, you can see blood cells flowing through using 2-photon microscopy. It's really amazing. It's expensive too. The device costs it's about $320,000 for the entire setup. Okay, so it's Now we're gonna talk about it a little bit more. We're gonna talk about electron microscopy. What you have to know generally for microscopy: method, the brief description, and application. I don't really care about how it works. So, for instance, if I ask you method that produces the 3D image by taking slices, you have to identify it as a confocal. Method that uses UV light to produce fluorescent image as well. It's kind of obvious, self-explanatory, okay? So be able to explain different types of microscopy in 1-2 sentences. Are we good? Okay.